Great pleasure to introduce today's speaker, Professor Popendorf. I'm sorry I did not do my homework on your short bio, so maybe
we went to the same grad program.
Yes, we did.
Yeah.
Joint program, and my team was called joint program. It's the best program that produced the best.
Yeah, we're a little biased, but yeah.
Releases.
And then after getting a PhD,
I was at Lamont Doherty.
At Lamont, another great center for oceanography, and after that you
And then I came here.
came here, so it was
That was great.
Okay, as you know, Professor Popendorf is coming for genuine promotion consideration, so today seminar is part of the process. Just puts extra stress on today's speaker. Okay, and without further ado, please, stage is yours.
Thanks so much. Yeah. So yeah, thanks everybody for being here. And as Igor said, this is sort of a dual function seminar. Roland's been having faculty give seminars here, kind of giving an overview of like who they are and what their program is. So I'm going to give a wide tour of the research that happens in my group. And then this is also an opportunity to tell you what I've been working on here in my run up to tenure. So I study marine microbial biogeochemistry, and we kind of have several prongs to my research. So I'll be telling you about the role of microbes in ocean phosphorus cycling, energy dynamics, and harmful algal blooms. And the pictures here kind of represent the typical workflow of science in my lab. We are largely seagoing oceanographers, so we go out on ships, sometimes in coastal oceans, and we collect water, usually using Niskins on a CTV rosette. And then oftentimes we manipulate the water to be able to look at particular microbial processes. So we'll do incubations, shipboard, and then we bring samples back to the lab, and we quantify all sorts of microbial and chemical properties. We use things like our shiny red flow cytometer, liquid chromatography, mass spectrometry, and all of this is in an effort to quantify the chemical processes that are mediated by these tiny little blue cells over here, the marine microbes. So as everybody knows, science is a collaborative effort. So I want to start out by thanking the fantastic members of my lab group. My current lab group is, most of them are sitting right here. So I have Dr. Daniela Maisel, who joined my lab 3.5 years ago as a postdoc. And I'm very excited we've gotten to keep her and promote her to an assistant scientist role. And I have 2 new PhD students, Claire Pandel and Sabrina Ufer, who joined my lab this fall. And are going to be here for their PhDs in the Ocean Sciences program. And I have a large cadre of fantastic undergraduates working in my lab right now. And because of their scattered schedules, I couldn't get them all in a picture. One day we will. But we have William Ostergaard, who's sitting here. We have Stevie Andrew, Allie Cook, Kayla Ellerby, Taylor Yagelinzer, and Allie Winkler, who all are keeping my lab running and doing a lot of science there. And a lot of the work that I'm going to present today is what's been done by the alumni in my lab. So this is everyone who's come through my lab in their time here. So I'm going to be presenting a lot of work by my first PhD student, Casey Lamker. You all on the faculty side and grad students as well are mostly familiar with her. She graduated in 2021. As she graduated, she got an NSF Ocean Sciences postdoctoral fellowship, and she took that to Scripps, where she's been working with Jeff Bowman. And then I had a master's student, Raquel Chennai, graduate this past spring. I had an MPS student come through my lab, Addison Testoff, and their names are going to crop up throughout the work here as well. And then I have had many undergraduates come through my lab, a lot of senior theses. And the asterisks are students who've co-authored, or have had first author papers that I've co-authored on. And so Haley Klaas, I highlighted, she's sort of a special story. She's going to come up later in the talk. She worked with me as an undergrad, done her senior thesis in my lab, and I've gotten to collaborate with her throughout her PhD. So I'm on funded projects with her and her PhD advisor, and she defends the end of this month. So it's been really fun kind of getting to be through with her through the whole process. And then I have a lot of other collaborators that I want to acknowledge as well. And again, there are students that are starred as people that I've co-authored with that have first author student papers. So I sit on a lot of people's committees, and for me that usually entails having those students work on analytical things in my lab. So I have a lot of great collaborators here at Rasmus, as well as across UM, and then highlighting other people that have contributed to this research here. And as always, very grateful for funding sources that enable all of this. So the things that we do in my lab are all looking at the role of marine microbes in ocean chemistry. And it's this strong sort of chicken and egg process. Chemistry controls microbes, and microbes play a fundamental role in controlling the chemistry of the ocean. So we're all about quantifying chemical processes, and we're just looking at the chemical processes that are mediated by microbes. And I'm going to kind of bin the research into three programs. We look at quantifying microbial energy, which we do by looking at ATP. We look a lot at phosphorus cycling, and then I work a lot on harmful algal blooms. So microbes are, by definition, microscopic. So I often start out my research, both in formal talks, but also informally, and people are like, so you do what now? Explaining why we look at microbes. They are tiny, so how could they possibly matter on a global scale? So when we look at the global carbon cycle, marine microbes fit in that marine biota. And in fact, they're so numerous that most of that marine biota is microbes. But when you look at that, it's 3 petagrams of carbon. And compared to other pools of carbon globally, that's really small. So again, it's easy to say, why bother? But the reason we look at them is because of the fluxes that move through that pool. So marine biota, microbes are responsible for transformations of more than 100 petagrams of carbon a year. And when you look at those fluxes compared to other fluxes in the global carbon cycle, those values are really large. And in particular, these microbes are important because they not only are responsible for large fluxes on a global magnitude, but they respond really rapidly to perturbations in the ecosystem. The average lifespan of a microbe is a day. So that means that as there's a sunny day or a cloudy day, the ecosystem changes. And in particular for things like an El Nino year and a not El Nino year, as ocean temperatures warm, as oceans acidify, marine microbes are responding really rapidly and have the potential to change the dynamics of the carbon cycle. And we know that they do because looking at paleoceanographic records, marine productivity has changed over glacial and interglacial scales, and that's played a critical role in changing the balance of CO2 between the atmosphere and the ocean. So the fluxes of microbes are why we're interested in them, and that's where most of our efforts go into quantifying the chemical processes within microbial systems. And again, when I'm trying to sort of motivate people about this, I often point out 2 facts: that marine microbes are responsible for half of global annual oxygen production. So you can buy a sticker that says, take a breath, thank the ocean. And then the other part of that, oxygen is produced by photosynthesis, and microbes are consuming CO2 in that process. And that microbial photosynthesis is what provides the potential for the ocean to be a carbon sink for atmospheric carbon dioxide. And at present, and since the last glacial maximum, that is how the ocean has functioned. And in modern anthropogenic era, that's really important because of all of these anthropogenic CO2 emissions that we produce, only half of them stay in the atmosphere. So that crazy increase in atmospheric CO2 that we see is the result of only half of the emissions remaining. The other half of those emissions are taken up by a balance of a terrestrial sink for that extra carbon and the ocean. And that ocean sink of CO2 is enabled in large part by microbial productivity. Temperature and saturation state and circulation are an intertwined process, but if microbes were providing a carbon sink, there wouldn't be such a large sink of atmospheric CO2 into the ocean. So again, our interest is really in looking at the fluxes of marine microbes, and they matter on this global scale, and that's kind of the approach that we take with the research in my lab, is thinking about things in terms of quantifying fluxes and being able to map them out to large basin-wide processes and Earth-scale processes. So those microbes, just giving you a close-up picture of them, again, they're really tiny. There's a scale bar on there that's 10 micrometers. So your average microbe is 1 micrometer in size. There's 10 to the 8th cells per liter. So this is like half a liter of water, and it would have 100 million cells, which is always more stunning thinking about it as 1 drop of seawater. Like 1 drop has 100,000 cells in it. And that's healthy ocean water. That's not, an algal bloom or anything. That's just average ocean abundances. And all of that, those microbes are transferring huge amounts of material from one component to the other, the atmosphere to the ocean, the surface ocean to the deep. And as they are, they're transforming it. They're transforming the chemical form and the reactivity and the fate, turning it from dissolved to particulate material in particular. And again, they respond really rapidly. So this is sort of motivates why we want to look at microbes. And we'll introduce microbes as 2 major groups to keep track of in terms of chemical processes. Phytoplankton are what are doing photosynthesis, so they take up CO2, they reduce that carbon, they turn it into organic carbon, and it takes energy to do that, and they're generating oxygen. So my students in classes always joke, like, I never get through a lecture without putting photosynthesis up on the board, so I'm giving you all the chemical equation for it. And it's just so fundamental to a lot of Earth processes. And respiration is just the reverse reaction of that. So we respire as people organic matter, we consume organic matter as our lunch, we take in oxygen, and we break it down. We oxidize that organic carbon back to CO2 to generate energy. And microbes are doing that as well. And the balance of these processes is what's providing the potential for the ocean to be that CO2 sink. So when we put this in terms of the ocean-atmosphere connection, phytoplankton are transforming that dissolved CO2 to organic carbon, and then primarily heterotrophic bacteria are responsible for this respiration, the reverse reaction. And sum total across the global ocean, it's net photosynthesis, which provides the opportunity for there to be that net CO2 sink of about 3 gigatons of carbon a year, 3 petagrams of carbon a year. And that's organic carbon, that particulate organic carbon that can sink out of the surface ocean and then provide the potential for the ocean to be that sink. And that is not evenly distributed around the globe. It's not evenly distributed with depth. It's not evenly distributed seasonally, and it's highly variable. And it responds to all sorts of parameters in the environment, the particular communities that are there, the nutrients that are available, the temperature of that environment. So those are the things that in my research we try and quantify, is what is the rates of production, what are the rates of respiration, what's the balance of these processes, and in particular, trying to quantify this in a way that can be predictive, and figuring out what are the factors that control that, how much of it depends on who's there. We measure how fast are they working, and then what controls the rates of that activity. So I always describe my research as trying to build a mechanistic understanding of these microbial processes. It's not just one sort of straight arrow for photosynthesis. It depends on the individual action of millions of tiny cells that each have different physiologies and adaptations to their environment and respond in different ways to changes in temperature and nutrients. So I often describe this as microbial dynamics. And I do mean that in the same sense that we talk about something like fluid dynamics, where the objective is to be able to build equations that describe the functioning of this system. So to be able to say, the organic carbon produced is a function of temperature and nutrients and viral abundance in this quantitative way. So we try and quantify things and be able to build these equations that would enable models that can run forward or backward in time and potentially providing predictive information for future carbon cycle questions. In the present state of ocean carbon models, microbes are the hardest thing to pin down. And you can see that when you look at people's predictions of the state of change of, say, temperature or primary production. Predictions for primary production are kind of all over the map on a very fine scale. So this is what we worked on. So within this, we're looking at this flux of phototrophic production and that heterotrophic respiration. And one of the key things in there is thinking about not only the flow of carbon, but a lot of what's controlling this microbial dynamics is the energy that the microbes need to do their metabolism. So we add on some arrows that respiration is providing energy and sunlight is as well. And that property of how much energy does an organism need to build its biomass is a really defining feature for how carbon and energy is going to flow through the ocean ecosystem. So we look at the rates of production, of biomass production and energy consumption to see how quickly things are spinning. And that's going to tell us a lot about how an ocean ecosystem will respond to changes. So we quantify this as a term as efficiency. And this is something we can describe for ourselves as well, right? It's like, of the material that you take in, of the lunch that you eat, how much do you burn for energy versus accumulate as biomass? And thank heavens, adults are very low efficiency creatures. We eat a lot, we burn almost all of it for energy. We accumulate very little as biomass. You're just replacing like fingernails and hair and sloughing off skin. So we're really low efficiency creatures. Efficiency varies enormously across life on Earth. And it's fundamentally a measure of how much resources required, what gets taken up for biomass produced. And most things need to burn a lot of organic carbon through respiration to get energy to support that biomass production. So when we talk about heterotrophic organisms like us, this is a nice carbon to carbon equation. And so we can talk about how much carbon do they take up, they're going to burn a lot of it for energy and build some of it into biomass. And the reason this efficiency is a really fundamental property to look at is that the fate of that biomass or organic carbon is going to be really different in the ocean. So if it's biomass, that's particulate organic carbon. That's what feeds the rest of the ecosystem. Phytoplankton are the base of the ecosystem. You need a lot of phytoplankton to feed some zooplankton, to feed some fish and some whales. And that's what can sink out of the surface ocean and provide that carbon sink. Whereas anything that gets burned through respiration for energy returns to that dissolved CO2 pool and it's going to equilibrate with the atmosphere. So these two processes are happening really rapidly in the surface ocean, and the balance of those processes is what's going to define the role of microbes in that carbon flow. So this idea of efficiency was established early on in the 1970s and 80s and defined for microbial and bacterial growth in the ocean. So this was at sort of the heyday of oceanography recognizing microbes as a really important role. So Pomeroy had this paper in 1974 that established this idea of the microbial loop. So at that time, there was a whole host of researchers, many of whom are still very active today, that spent a lot of time quantifying this efficiency, coming up with ways to measure bacterial production rate. And measuring that respiration rate is really hard. Chris Langdon is very good at it and can attest to how hard that is. So the ratio of that total carbon taken up, the amount that goes into production, is what we define as efficiency. So production rate divided by total carbon uptake is our efficiency. And measurements were made, and we know there's a range, and it's ballpark, most material gets burned for energy, ballpark 90%. But this is where the variations matter. It ranges from about 1% goes to biomass to 30% goes to biomass. And in a given ecosystem, that's going to be a huge swing in where carbon is going. And we don't have a great handle on what controls that growth efficiency. And one of the reasons is that it's really hard to measure that respiration side in particular. So we don't usually do this at broad scales or over a lot of temporal time points for looking at how things vary in response to changes. And in particular, right now I'm describing this all for bacteria who are heterotrophs like us, and at least it's a carbon-to-carbon question. They take up carbon, and that becomes their biomass and their energy. When we look at phytoplankton, this gets even more complicated because they also have a sense of efficiency. You can quantify efficiency, how much biomass gets produced, but the energy fueling that production comes now from both respiration and from light. And so now your denominator has apples and oranges in it. And so it's really hard to quantify this flow of carbon and energy the same way through phytoplankton as we do through bacteria. But it's the balance of these two groups that's really defining the function of the ocean in the carbon cycle. So we wanted to be able to take a look at microbial growth efficiency as this production relative to the resources that are needed, incorporating both energy and light, and be able to quantify this efficiency. So a lot of my PhD was dedicated towards developing new ways to measure bacterial production rates. It took a long time to publish that, so it came out fairly recently. And the upshot of that was that bacteria grows far faster than usually assumed, probably about 10 times faster than what was established in the 1980s. But what I'm focused on here is getting a better quantification for the energy side, in particular the way that we can look at phytoplankton and bacteria. So I thought it would be a good idea to have that be the first project for my first PhD student. Just come up with a new method to measure energy in marine microbes. So that's what we did. So Casey Lanfer developed a method for looking at that energy flux rate. We do it looking at ATP in marine microbes. And then we employed it in a lot of different settings and looked at how that energy flux changes in response to temperature and across different ecosystems. So the idea of using ATP, it's adenosine triphosphate. It's a ubiquitous energy trafficking molecule for all life that we've found so far on Earth uses ATP, including us. So it's this really high energy compound. And what cells do is that when energy is available, say from respiration, they add phosphate groups onto that adenine. And the bonds between those phosphate groups carry a whole lot of energy. So it's this little chemical battery pack that then floats around the cell. And whenever a metabolic process like carbon fixation needs some energy, it can break off that phosphate bond, and it supplies energy for all the metabolism that cells do, for building proteins, for replicating DNA, building new membranes. So in cells, ATP is constantly having phosphate groups added on and having them broken off. And so if we can measure how quickly that phosphate is getting added on, that gives us a measure of the energy flux in a cell. So that's what we do in our lab, is measure how quickly phosphate groups get added onto ATP in marine microbes in the ocean. So, and again, the really powerful part of this is that ATP is a widely used form of energy. So it gets generated both from phytoplankton capturing energy from light and from any respiration. Both of those will generate ATP. So now this gives us an apples to apples denominator for looking at efficiency, and it'll work for phytoplankton, it'll work for bacteria. So again, we're going to try and quantify that phosphorus group being added on to ATP as a way to quantify energy flux in the environment. So the way we do this is that we add in radio-labeled phosphorus. We take a sample of seawater and we spike it with P32 labeled phosphate. So that's radioactive, and adding that radioactive material acts like adding a dye to a river, where if you add dye to a river, it's easy to see where the water flows. Radioactivity is really easy to measure, and we can basically track where that phosphorus goes in a really quantitative way. So that phosphorus is going to get taken up and it's going to get added onto those adenine groups. So then we filter out all the microbes, we extract the ATP out of their cells, and then we purify the ATP and measure how much radioactivity got incorporated in a really short amount of time. So again, the conceptual idea for this was introduced in the 1980s in kind of this early heyday of microbial oceanography. Dave Carl and collaborators, Bossert and Holm Hansen, laid out the conceptual framework for this. And they introduced a method to measure ATP phosphorylation rates, and it used thin layer chromatography, which was the best we had in the 1980s, and it is so laborious and hard to do successfully that it fails most of the time, even by people who are trained in Dave Carl's lab. So it hasn't gotten applied very much. So you can look, and there's a lot of the value of this measurement is kind of clear from the 1980s, but there's very few applications of it. Because it's really hard to do it successfully, particularly in an open ocean environment. So our goal was to make a method that we could actually apply at scale, robustly, and be able to measure across a transect or in a time series type setting. So this is a year or two of PhD work for Casey in one slide. This is her method that she developed, where again we take seawater, we spike it with radioactive phosphate, filter to get all the microbes, extract the ATP in boiling tris, centrifuge to separate things out, and then you can freeze it at that point. This is great, because you can do all this first part on a ship, bring all your samples home, or we can send an HPLC out on the ship, and then we use liquid chromatography to purify that ATP. And that's the critical step that transforms it from what was introduced in the 1980s into a scalable method that's robust and that we can apply really broadly. So this is what that chromatography looks like. You use a chromatographic column to separate out all of the compounds that were in those cells and purify ATP. You can also get ADP and AMP. And then we actually physically collect what's coming off the column, and that the amount of radioactivity in that tells us how quickly ATP was being phosphorylated. So we spike for a really short amount of time, measure how long, purify the ATP, and measure its radioactivity. So it's this HPLC radio labeling method for measuring ATP turnover, which gives us energy flux in the environment. And again, the key part of this is that we can really now do this at scale. You can employ it for large data sets. We can measure across transects, in time series, or in large manipulation experiments. And now we've done all of those things across the work that Casey did in my lab and what she's carried on in her postdoctoral work. So the first time that she employed this method was on a ghost ships cruise across the South Pacific. So this was the P6, the second leg across the Pacific Ocean, so going from Tahiti over to Chile. Working within the constraints of ghost ship, we actually can't work with the radioisotopes, so we were just getting ATP concentration with this method. But here too, this is a large data set of ATP relative to what had previously been published. So here you can see the concentration of ATP across this transect in a cross section from the surface down to about 200 meters. And there's some distinct patterns across that transect. And so our question in this work was, how does the amount of ATP compare to other measures of biomass? So what you see on the left is a bunch of different measures that we made for biomass. We measured particulate phosphorus concentration, the total number of cells, biovolume. Those two have patterns across the transect. Some of them reflect slightly different patterns. And so our question was, what does the ratio of ATP to biomass look like? Are there patterns in how cells are allocating their resources and how they're using energy? Whether they have a lot of energy storage capacity or a little bit of energy storage capacity, is that systematic across the transect? So this is ATP concentration relative to each of those measures of biomass. And what you can see is that there are distinct patterns across that oceanic transect, and they map on the patterns of nutrients and oxygen. So we binned our samples across the transect based on the nitrogen to phosphorus ratio in the water and the oxygen concentration. And then the samples across that transect really kind of fell out into these four distinct groups. Ones that had higher nitrogen to phosphorus ratios, and in particular then ones that had this low oxygen concentration. And when we look at that ratio of this energy storage compound, ATP, compared to biomass, there's patterns across those regimes, different ecosystems that we can see based on nutrients and oxygen. So for cells that were below the euphotic zone and in this higher NDP regime, they had significantly more ATP per biomass. They're putting more of their resources into energy storage. And within that, we could break out this group of samples over here that were in the low oxygen area in the eastern Pacific upwelling zone, had really high levels of ATP to biomass, significantly different even from the other deposition or sub-euphotic zone microbes. So this shows us that there are some really different strategies of microbes for allocating their resources towards energy versus the rest of their biomass, and it maps on to physicochemical properties in the environment, onto nutrients and oxygen. So this is some tantalizing evidence of like the power of looking at energy and biomass as a way to kind of understand how things are flowing through the ocean. So we did this on one ghost ship cruise, and I've been really fortunate to be able to, through other PIs that are here at Erasmus, that are in the ghost ship program, have the opportunity to send students out. So Casey went on the ghost ship cruise, and this was some ancillary science. So we put in kind of a separate proposal to do these add-on measurements, and falls into this category of synergistic opportunities on ghost ships. So we did that successfully in the Pacific. We were lined up to do it in the Atlantic in March of 2020, which got called off. So she was going to complete a lovely 3-basin set in her PhD of Pacific, Atlantic, and Indian Ocean. She went to the Indian Ocean as part of the Kudos program and made similar measurements. We've gotten some funding independently from NOAA for that synergistic opportunity. So we retooled, but Claire is hopefully going to be going out on an Arctic cruise next summer. So we really appreciate the opportunities of being involved in ghost ships. And part of this then is also getting to present this work through ghost ships is really trying to make a point about the synergistic opportunities of being able to expand beyond the core parameters in ghost ships. So we are one of a number of PIs that do this, where we kind of help out with the core parameters and add on other biogeochemical measurements. So ghost ships has been kind of promoting that internally and funding agencies, and that's been part of building a lot of community momentum towards more biogeochemical international ocean sampling programs. So there's this program called Biogeoscapes that's been in sort of the town hall planning workshop phase for a couple of years, and is hopefully coming towards fruition. So the Biogeoscapes is looking at ocean metabolism and nutrient cycles on a changing planet. So we're at the stage of having a science planning workshop, so I'll be participating in that in November of this year. So hopefully more large ocean transects, and with the opportunity maybe do more of the rate measurements out there. So again, with this ability to look at energy, we're really interested in looking at these, the flow of material and energy through phytoplankton and through bacteria. So looking at the balance of production rates and energy consumption. And one of the things that we were really interested in looking at was how we think this is going to change the balance of energy needed for biomass produced in a warming ocean. Because there's a lot that's known about metabolism and temperature dynamics. And if you take any organism and you warm it up a little bit, metabolism is going to go faster. And that's just the principles of enzymes. It's defined by things like the Arrhenius equation and the Van Hoff equation. And the oceans are warming. So ecosystems are going to change. And there was a really interesting article that came out over the summer that it's the first observational evidence that subtropical gyres are greening. They're turning green observationally faster than we predicted. The ecosystem is already changing. So this was a Stephanie Dukowitz, Stephanie Henson paper. So we were interested in checking this out. So this falls into the category of work in my lab that I refer to as experimental assessment of climate change. So during deep COVID, when we couldn't go out on oceanographic cruises, we turned to a really relevant place for looking at temperature change here in our coastal ecosystem and where we sit right at the origin of the Gulf Stream. So conducted an incubation experiment where we took ocean water and then we elevated the temperature. And so we do this set up incubators quite often on the dock. You may have seen us out there. So we pump flow through seawater to be able to maintain ambient temperature and have our phytoplankton in ambient sunlight. But then we were able to modify the conditions. So in this case, we were trying to do sort of climate change relevant temperature increase. We also love using household tools for science, so we were using an immersion cooker that you might use for like sous vide steak. It did a great job of warming up our water 4 degrees C above ambient temperature. So we were doing these multi-day incubations to see how microbial energy flux and nutrient flux were going to change in response to temperature. If they were going to change in concert, if everything was going to speed up, or if they were going to start burning more energy for the amount of nutrients they took up, or vice versa. So we did this in the winter and in the spring. And you can see kind of the average temperature ranges in each of those, so January and August. So we were focused on the energy flux and nutrients, and then measured a whole host of biomass parameters and nutrient parameters to get kind of a full picture of what was going on in the ecosystem. So looking at that energy flux and that nutrient flux in the summer and in the winter, in our 2 different conditions, we saw changes over time and some systematic patterns. And there's a lot going on here, so I'll break it down with one of the comparisons we were really interested to make was on that first day, before we had kind of subjected these microbial communities to being in a bottle for a while in their altered conditions. If we just look at winter and summer in Miami water, it's conveniently about a 10 degrees C difference, 24 degrees C to 34 degrees C. And I should point out that when I presented this work at like national or international conferences, I blow everybody's minds with how warm Biscayne Bay is in the summer. And they're always like, that's ambient? And then that was even before our like record-breaking temperatures this summer, right, where we hit like 38 and I think up to 40 degrees C in some parts of the waters around here. So at the time, we thought we were being, really hitting the high bar there with like a few degrees above 34, but turns out we really underestimated the environment. But nonetheless, that 10-degree temperature change is a really useful parameter to look at for metabolism. There's this idea of the Q10, like what fold increase do you see in rates with the 10-degree temperature change? And based on what's known about protein kinetics and enzymes and changes in temperature, most metabolism expected to increase at a 2-fold rate with a 10-degree increase in temperature. So that's pretty much what we measured for our energy flux rates. In the summer, when it was 10 degrees warmer, the ATP phosphorylation rates, our energy flux rates, were about 2 times faster than in the winter. And when you look at the nutrient flux, the change was much larger. There was a 10-fold change, a 10-fold increase with warmer temperature in the phosphate uptake rates. So that was an interesting finding to see that was the direction that went. But the reason that we did this as a manipulation experiment rather than just a seasonal observation experiment is that a lot of things change winter to summer. The nutrients change, and the community that's there is very different in the winter to summer. Water's moving through, and it's a dynamic system. So that's why we looked within our modified conditions. So we can look at the last day of the experiments. This is like day 3 in the summer, day 4, to really look, across our induced temperature changes, how did nutrient fluxes change? And what you can see is across our winter and our summer conditions, there was this predictable increase in nutrient flux with an increase in temperature. And it was a pretty monotonic increase with temperature across all 4 of our conditions. So this, again, getting at that idea of like dynamics and being able to put some equations on how we expect things to change, we were really excited. We're like, numbers, quantitative values that we can potentially build predictive models from. When we look at the energy flux, in the winter, it also had an increase in energy, and it was in fact a larger fold increase in the energy flux by the 4th day of the experiment than the nutrient flux. So that changed, we saw a larger change in energy when we had the same community there. But notably, in the summertime, our rates were really low. We don't see at all the same pattern with that like monotonic increase. And this was really interesting to us as well, because what that indicates is that potentially we are at the edge of the optimal range of temperature for these microbes. Every organism has sort of a habitable zone for temperature where rates will increase up to a certain point, but then based on the physiology of that organism, there will be a steep drop-off in production or health past a certain temperature point. So this was the sort of thing that bears more investigation in the future. So we got a lot of really interesting quantitative results from this, and have plans to continue on with a lot of experimental work looking at properties like this in this and other ecosystems. So again, broadly, we really want to be able to quantify these rates of production and energy and quantify them in a way that we can put together dynamic equations about how production and energy rates of these different microbial groups relate to temperature and light and oxygen and nutrients and the community that's there, and build this mechanistic understanding. So one of the places we're going to do that is that we're going to be going out and measuring microbial growth efficiency, so that's that energy and production rates, in the California upwelling system. So this is, we designed this grid sampling that we're going to be doing off the coast of California because it spans a lot of different physicochemical properties. You have upwelling, so we've got cold temperatures and warm temperatures. We have high oxygen and low oxygen, big ranges in nutrients, including ranges in nutrients within the euphotic zone, high nutrient areas and low nutrient areas. We're expecting we'll get ship time in 2025, so this is a funded NSF project with 21 days of ship time, probably on the Sally Ride, where I'll be serving as chief scientist, and I'm collaborating with Jack Bowman at Scripps and Sherry Palacios at Cal State Monterey Bay on this project. And the idea is that across this grid, we're going to be collecting huge amounts of data, ballpark 500 data points for 15 or so or more different parameters, so depth profiles at 36 different stations. And the idea of this is to be able to really build this into some sort of advanced statistical models. So Jeff Bowman specializes in building statistical models with machine learning and uses random forest processes, and to be able to really identify drivers for this system. So again, getting back at that idea of dynamics, identifying where and when is temperature a driving force for variations in nutrients, and what's the relationship between nutrients and carbon flux and energy flux. And one of the things I'm really excited about is that built into this program, we're recruiting 5 undergraduate students from the Cal State University system to take them out on the research cruise. We're kind of running our own mini RAU program. So the idea is enabling them, they'll be part of the core sampling, but also do their own independent research projects. And I was really delighted to be able to actually fund this in the broader impacts in a real way. So we have help from Cal State through their COAST program for trying to really design the recruiting well with a lot of thought to help bring in diverse students and make it a really inclusive process, and use this to kind of broaden participation in marine sciences. So working with undergrads at sea is something that I love to do, and I've put a lot of time and energy into here. So one of the ways that I do this is through the Flotsam program. So Hillary Close and I organized sort of the initiation of this program in 2019, when fantastically the school provided funding to take undergraduates to sea. So Hillary and I kind of set this up that first year in 2019. Hillary came up with our awesome acronym, which is the Florida Ocean Time Series by undergrads at UM. So we take about 20 students a day out on the Walton Smith to the Florida Straits, about a 2-hour steam from here at Erasmus, and we sample a whole host of physical and chemical and biological properties. So what I did that first year was kind of set up our where we were going and what we were going to sample and how we were going to sample for it, and kind of this rotation of activities, so everybody that goes out gets to get experience with all these things. So I chief side for the first set of cruises, and as most of you know, Jim Happel has been fabulously running this program for the intervening years, and it takes a huge amount of effort to organize the people. We have hosts, most of you in this room awesomely have been out to sea, so thank you for coming out and teaching our students. So they really we get such good feedback from this. They learn, some of the comments are, they learn more in a day at sea than in a year in their classes, because they just get to see everything coming together. So this has been something that I've loved doing, whether or not I'm chief science or out on the ship. Every time we go out, I'm loading the gear, I'm training, I do pre-cruise meetings with the students. I did that yesterday, and then training the people that are going out to sample, and then I archive the data in the samples. So that this gets used in a lot of classes. Every undergrad class I teach, we use data and samples from the Flotsam cruises, and a number of other people do as well. I know Mo's used some of the CTD data in his ocean data class. So tomorrow we're going out. I'll be loading the ship this afternoon along with Jim and a number of other people. It'll be our 21st cruise. We've taken close to 400 students to sea, well over 300. And with this, we've been doing this for about 5 years now, in the fall and the spring. So this is some valuable time series data that we're building. So I have a pending NSF career proposal that, if it gets funded, will provide funding and critically student personnel to really formalize that time series. Right now I'm sitting on piles of data that I don't have time to do anything with. So if any of you have eager students or plans yourself, let me know. We've got samples and data, so much of it. And hopefully we'll get some funding to continue to formalize that, and also kind of broaden out our research at sea program for the undergrads. So I'm going to do a quick overview of 2 of the other prongs of our research. We work on phosphorus cycling, and then I'll touch on harmful algal blooms. So again, all this kind of fits in the role of microbes in ocean chemistry. And the reason we focus on phosphorus, it's one of the two major nutrients that every microbe needs. They need carbon and nitrogen and phosphorus. In the ocean and in phytoplankton, this occurs at this characteristic ratio, molar ratio of 16 parts nitrogen to 1 part phosphorus, and 106 parts carbon. This is known as the Redfield ratio. So phosphorus is the least of those, but it is also, because there's this back and forth control between microbes and chemistry, it's also in least supply in the ocean. So this is a picture of global surface phosphate concentrations, and you can see where we are here, near the North Atlantic, is the lowest phosphate concentrations in the global ocean. And that matches with patterns of global circulation. Surface water in the North Atlantic has been circulating on the surface for a long time, and microbial activity has just been depleting away that phosphorus as it goes. So by the time it gets here, it's at very low concentrations, and that is often a limiting factor for microbial productivity. So whether it's the ultimate limiting nutrient or one of the limiting nutrients, it's often a lever that helps control productivity, and one of the fundamental things to look at for those dynamics of, if nutrients are controlling productivity, phosphorus is an important one to look at. And when we work in oligotrophic ecosystems, looking at phosphorus is fun and fascinating because microbes have a diverse array of adaptations for getting more of it or using less of it, and each microbial species specializes in a different way. They have all these niche adaptations which are really interesting to look at and then play out in these really broad patterns that lead to things like the oligotrophic oceans being dominated by cyanobacteria. So we know enough about a lot of these dynamics to get basic patterns like cyanobacteria dominating in gyres. That comes out in our current biogeochemical models. So that's a lot of work from like McFarlane's has kind of led the charge on that. But there's a lot that we still don't capture in our biogeochemical models. So we work on phosphorus as trying to hone in on those details and look at those levers. So again, we're looking at fluxes, quantifying a lot of rates of inorganic phosphorus and organic phosphorus going into different microbial groups. So we've got phytoplankton and bacteria. I've done this work mostly in oligotrophic areas because that's where it's most impactful and interesting. So in the Sargasso Sea, coastal waters here, and the Gulf of Mexico. And we look at a lot of different microbial groups because they each play different roles in those carbon and energy cycle processes. And use tools like the ones that I mentioned, flow cytometry, we do more isotope rate tracing, adding in radioactive phosphorus, and then liquid chromatography for distinguishing different biochemicals. One of the projects that we did with this was funded from my first NSF project that was active when I was just coming here, where we went out and added radiolabeled phosphorus to use cell sorting flow cytometry to actually separate out environmental sets of these different microbes and look at where in the cells the phosphorus goes. And this was before we had our method for ATP. So here we were looking at phosphorus flowing into DNA and lipids and polyphosphate, which are some really critical biochemicals. So what you're looking at here is the distribution across different microbial groups. And what you can see is that the pattern of distribution is different for bacteria versus phytoplankton. They've specialized in different ways. They use that phosphorus differently in their cells. And the reason this matters on a broader scale is that each of these is little packets of particulate phosphorus at great abundance, packets of particulate material at great abundance in the ocean ecosystem, right? It's 100,000 cells per milliliter for bacteria. So there's all these little nuggets of particulate material at different sizes and abundances, and the fate of that material, whether it's grazed or aggregated, is going to carry different chemical signatures and is going to feed the ecosystem different ways. So part of the upshot of this is heterotrophic bacteria are actually really phosphorus rich compared to phytoplankton. They've developed a lot of strategies to win out at getting that phosphorus, and they have a higher stoichiometric ratio of phosphorus compared to the other cells. So one of the other things that we look at for thinking about phosphorus cycling is where these pools of phosphate are coming from. What's resupplying phosphate to the surface ocean? Globally, it's upwelling in a short-term sense, but like where does it come from in the ocean to begin with? It comes from riverine sources and a lot of aeolian inputs, so aerosol inputs. So I have the great fortune to share my lab space with Cassie Gaston, who's a fantastic atmospheric chemist, and I've gotten to collaborate with her and her grad students on looking at atmospheric deposition of phosphorus. So working with her grad student Anne Barkley, we were looking at the deposition of African dust to the ocean. So this is the west coast of Africa, and you can see a satellite picture of these huge storms of dust, which of course we're well aware of here in Miami, because seasonally in sort of August timeframe usually is when that dust peaks. It gets transported all the way across the Atlantic, and when we have events like what was termed the Godzilla dust storm, it'll actually be like a fine film on your windshield. So dust is famous for carrying a lot of, say, iron. This was John Martin, his iron hypothesis, like give me a tanker of iron and I'll give you an ice age. So it brings iron and it also brings phosphorus. So one of the things that Anne was looking at was quantifying how much of that mineral phosphorus is soluble. The answer is about 5%. But what was the real exciting takeaway of her paper was looking at other types of aerosols, specifically biomass burning, which also gets transferred from Africa with a different seasonal pattern. So it's getting deposited at a different time in a different pattern of upwelling in the ocean, and it's far more soluble. It's 10 to 20% of that phosphorus is soluble. And the net effect of that is that biomass burning may supply up to half of the phosphorus that's deposited annually on the ocean. So it was sort of a really under-quantified source of phosphorus. So Cassie's lab has carried on this work. So I've gotten to work with Hope Elliott, who's one of our ocean sciences students, and looking at the impact of even more varied aerosol materials. And that's, we got to look at that Godzilla dust storm. So this was really high deposition rates of dust. And compared this to another atmospheric event that occurred around the same time. There were large volcanic eruptions in the Caribbean that generated a lot of volcanic ash that landed on the ocean. So here, rather than looking just at the chemistry of those particles and their solubility, we looked at the microbial response. So this was, working as an oceanographer, working with like volcanic material was really exciting for me. So the day that we set this up, we were down on the dock and we collected ocean water, and then we added in volcanic ash from this eruption, the Lesser Freer eruption in the Caribbean, and added in dust from the Godzilla dust event, and looked at the impact of that on phosphorus drawdown and phosphorus uptake rates. And what we found, part of what's really new in her work, is that ash has even lower solubility phosphorus than dust. And that has been wildly misrepresented in the literature so far because it's not measured very often. So it's usually assumed to be 100% soluble, and we measured it as being 1% soluble. And despite that, adding ash, volcanic ash, had a larger impact on phosphorus uptake rates by marine microbes. So we added in some ash, and what you can see here is the uptake rates immediately after we added the ash. So like day one, take a jug of seawater, add in leached ash, shake it up, and then measure the phosphate uptake rates. And they were really high in response to the ash. So that's an exciting indication that volcanic ash is likely to have really significant effects. And this led to, so this is a chapter that I put in prep. I think by the end of the day it might actually be in review. So this has led to some exciting, further questions of with such low phosphorus solubility, what else is going on in the other trace metals? So this is some of the phosphorus work that we've been doing in my lab, and with each new thing opens up new questions. So more of that to come. We might be looking at some other biomass burning like wildfire additions, but Hope and I will hopefully be going to sea and measuring some microbial incubation similar to this in some more far from shore ecosystems. So with a little bit of time I have left, I want to tell you about the last part of what I do, the last major sort of research program in my lab, which is looking at harmful algal blooms. So this fits in the same realm of thinking about the role of microbes in chemistry, how they respond to nutrients, and here in particular, the biochemicals that they produce, notably toxins. So we term big blooms or proliferations of phytoplankton harmful when they produce toxins. That's what makes it a HAB, or a harmful algal bloom. There's all kinds of varieties of organisms that produce HABs. So here in Florida, of course, we get red tides, and that's a dinoflagellate. Carenia brevis is what generates red tides. All these different types of species produce a wide array of different toxins. Each of those toxins has a different physiological effect. So some cause things like paralysis. Saxitoxin is an extremely strong neurotoxin. Other things like ciguatera produces extreme gastrointestinal distress. And how people are exposed to those toxins also matters for the effects that they have, whether people are ingesting that, if they're eating shellfish, that bioaccumulated toxins, or they're drinking the water, if they're getting a dermal exposure because they're swimming or working in a HAB setting, or if they're inhaling aerosols of these toxins, which is the hardest to avoid exposure route. So this is where I get to loop back to working with Haley Ploss. So I've gotten to continue to work with her and her PhD advisor Hans Perl at the University of North Carolina. And when we look at HABs, they're a global phenomenon. They are naturally occurring, but their incidence has been increasing over recent decades due to a combination of climate change, warming waters, but especially human activity along coastlines. Adding nutrients increases the incidence and sometimes the severity or the frequency of HABs. So here in Florida, we are blessed with an abundance of HABs of all varieties. So this is what got me into HABs when I started here. So there's a lot of research about harmful algal blooms going on here. So I focus particularly on freshwater, blue-green algal HABs, so these are cyanobacterial HABs or cyano-HABs. Within that, what's most abundant here in Florida is microcystis, which produces the toxin microcystin. So I have projects going on in my lab working on Florida, which is what I'll tell you mostly about here, also field experiments in North Carolina and in California. And within the realm of cyano-HABs, I focus specifically on those aerosolized toxins. So it's sort of the exposure route that we know the least about. And I work in the intersection of in the environmental sciences and public health. So for this project that I lead in Florida, it's funded by the Florida Department of Health. And this grew out of a ULink project. So I want to give a good shout out to ULink. So Brian and Cassie were part of this initial ULink project. And this is our team now that's funded by the Florida Department of Health. We are a large and diverse team that span from here at Rasmus, the College of Engineering, and a lot of people at the School of Public Health. So our program is known as the Diversity and Innovation in Screening and Prevention of Exposure over the long term to harmful algal blooms, which gives us a nice acronym to spell to HABs. So we're funded by the Florida Department of Health, and working with state funding means that we're on an annual cycle of renewal, which is nail biting every May as the budget by the state gets approved. But I'm very happy to say that we're in our 5th project year of funding, continuous funding. So we've really gotten to grow this project. It's been a lot of funding over that time, over 1.5 million. And this is a community engaged science project. So we're getting environmental data and health data from community participants. And we have over 100 people across Florida that are part of this now. So what this looks like is that we are recruiting people that are exposed to HABs either because of where they live or where they work or where they recreate, if they walk their dogs along areas that have a lot of HABs. And then we're also trying to recruit visitors. If anybody has suggestions, this is the hardest to recruit group. We're like, you're here to tour Florida. Want to do some health samples about the harmful algal blooms you're exposed to? So we're mostly trying to use our residents and workers, if they have visitors coming, to talk to them about the study. So from each of these participants, we get demographic and survey information about their exposure and their symptoms, their underlying health. We get pulmonary function information, and then we have them collect water samples from the areas that they're being exposed to. They do microbiome swabs, so that's like their own microflora of microbes. We have them do nasal swabs that we measure for microcystin. So this is one of the main ways that we get a measure of aerosolized toxin exposure. And notably, you can see we began recruiting in February of 2020. This took a real turn when COVID hit. We could no longer go see people in person. So we now run all this remotely. So I'm really happy to say we got to continue running it throughout that time. And people are really good at collecting nasal swabs. Turns out they don't need much instruction on that these days. And if we can go see people, we can also get a blood sample. So to recruit these people, we work with a lot of community partners across Florida. So these are all organizations that we've developed relationships with and meet with and speak to their board and their community members. So I do a lot of pitches to community organizations. And our Addison Testoff, who worked in my lab through the MPS program, is now our program manager. So he's been working with this project for 4 years as a fully employed program manager. So he does a lot of our participant recruitment. We get a lot of press from this because harmful algal blooms are a very public issue here in Florida. And I really want to give a shout out to the Rasmus Communications team. They've really helped facilitate this. Talking to the press was very new for me when I started this, as was working for people, so it's been a huge learning curve. And as the lead PI on the project, I get a lot of calls. So we've talked to the press, which really helps with our recruitment. This Florida Today article came out a few weeks ago, and I think we've had at least 10 people contact us and join the study. That's always like we get contact, and then we talk to them about what the study entails, and understandably many people are like, okay, cool, thanks. But we've had at least 10 people actually join the study as a result of that one news piece, so thank you, Florida Today. So we, when we're looking to recruit people, we also do this through postal mail. It turns out people read their physical mail. We send out flyers to people around Lake Okeechobee and the connected waterways at the St. Lucie estuary and the Caloosahatchee River. This is sort of a heat map of where we've recruited people so far. So we have an ever-growing amount of data, and one interesting thing in this is figuring out when to kind of pick breakpoints to publish, because it's an ongoing study, which is really different for me than like doing a field campaign and being like, now we know what we know and we shall publish, and here we're like, we are still going and getting new people and new samples. But we have 2 papers from the cohort study that we're going to try and publish this fall. We also have worked on how to prevent exposures. So we did some experiments, some laboratory experiments, again facilitated through Cassie Gaston's atmospheric chemistry expertise, to see if people can minimize their aerosolized exposure using face masks and air filters. And the great answer here, which I love sharing with people, is that yes, we can minimize exposure. So these are lab experiments. And then we've used that, again, they set up of lab experiments with cultures and generating aerosols to see if we can use silicone wristbands as a way to monitor for aerosolized exposure. So silicone wristbands get used as exposure tools for a lot of other things, like PAHs, polycyclic aromatic hydrocarbons, that firefighters get exposed to a lot of. So there's a rich history of using them for other chemical compounds, but nobody had tested them for HABs. So we tried this out. This was the master's thesis, one half of the master's thesis of my student Raquel Chenet. And we're delighted to see that they work. They capture aerosolized toxins in a quantitative way. We can extract them off and measure that. So we've started deploying those in our kits that we send out to our participants. So now we're getting some coming back in from people just in the last few weeks into the lab. So we have a video about that face mask and air filter study, this sort of a public service announcement about how you can protect yourself from HABs, and some publications that have come out of that. So that wraps up the rapid tour of different research programs that we have going on. And again, we're really excited about being able to quantify chemical processes that are mediated by microbes in this meaningful way that we can build kind of this dynamic mechanistic understanding of what's controlling the flow and map this back out to these big picture processes of what are microbes doing in terms of the flow of carbon and energy in the ocean. So I'll put up my collaborators one more time, many people to thank. And with that, I'll take any questions.
Thanks for the really nice talk. Amazing note. I got a little confused about the ash uptake of the phosphorus portion.
Mm-hmm.
So is the soluble phosphorus the part that the ocean cares about, or the microbes?
Yeah, it's the soluble part is what the microbes care about. Microbes are osmotrophs. Everything they take up is dissolved. So it's the soluble part. But one thing that I didn't get to dive into too much, when we measure the soluble phosphate, we're usually measuring just the inorganic component, the phosphate. There's also organic phosphorus, and that's actually one of the other things that Hope was quantifying here was the organic phosphorus fluxes. We were measuring alkaline phosphatase activity. So that's another layer of detail. So yeah, that's great. Like the results were really surprising because microbes care about that soluble part, which is a small part of the total phosphorus coming from ash. And nonetheless, ash leads to a really rapid uptake in phosphate.
So is that because there's something else?
That's what we think, yeah. So this is where then we had some great discussions about the other trace metals that are abundant in volcanic ash. And particularly for that dissolved organic phosphorus uptake, that's facilitated by enzymes that usually need a little bit of cobalt or zinc or magnesium to do that dissolved organic phosphorus uptake. And with that alkaline phosphatase, this gets into a lot of nitty-gritty details, but it's usually an extracellular process. So if they get cobalt, they can make more enzymes to lyse the organic phosphorus out there extracellularly. So we might see this, and what we're measuring would track a boost in extracellular hydrolysis of organic phosphorus enabled by trace metals from the ash. If that makes sense. So that's kind of that was one of the hypotheses that came out of this work, is that ash gives a boost because of other trace metals that are really abundant in ash. So another part of what I was doing selected pieces of the story, she was looking at the iron chemistry, like iron 2 versus iron 3, which also defines its solubility and activity for microbes. And then what we would want to do next is looking at the trace metal chemistry of the ash and dust and biomass burning.
Thanks.
That's super fascinating, and I just didn't know any of this, so thank you for that. Just my impression is you start with the budget of particles big enough to sink for reasons of global climate change or something, very human agenda. You bring that to ecology, discover, oh, that's tied up with energy, okay, bulk energy, bulk matter. Oh, you know, it turns out the real driving dynamics are not driven by bulk energy and bulk matter, they're driven by these trace nutrients and phosphorus. Okay, and then phosphorus, when it goes wrong, you get these pathological cases, and now you got all this research about the pathological cases. If you were interested in marine microbial ecology, would you approach it from these twin overlays of a waste product over here and a pathology over here? Or are there opportunities going missing because all our research is justified by pathological crises and a global climate agenda?
Yeah. Well, so I'm happy to say that there is a really active microbial biogeochemistry research community that comes at it from the chemical perspective. So usually I define myself as a chemical oceanographer, and that's why I start with like the big picture, like carbon processes. And I look at all those other, the phosphorus dynamics and things really as like what's controlling how quickly things spin. But we do a lot of measurements that I didn't go into details to really just quantify the carbon flux. So there is, thankfully. So NSF still funds a lot of kind of big picture, quantify the fluxes measurements, and things like the Simons Foundation. But there's separate pools of money for things like the harmful algal blooms and the interactions with public health. But yeah, there is like kind of big picture flux support for that. So those things like the biogeoscapes programs is really, it's a lot of chemical oceanographers that approach it from kind of that big picture perspective. Does that answer your question?
Yeah, maybe spotlight the ecosystem for its own interest, rather than for a human impact over here and a human impact over here.
Yeah.
Are ecologists well enough funded, or is all the money going to biogeochemists?
Right.
Is there something
Well,
like that?
I don't know. I guess I usually do come at it from that like flux biogeochemistry perspective. Microbial ecology sits kind of more squarely within biological oceanography and like within NSF. That funding is far more competitive.
There you go.
Yeah.
That's what I'm getting.
So actually, I usually apply to chemical oceanography, and then they chat amongst themselves and decide if they're going to pass me over to biological oceanography or not. BioOcean keeps funding me, sometimes jointly with ChemOcean, but they've decided that I sit somewhere in between the two, which is working out funding-wise, so I don't say no. But
so that figure that you had of like the nutrient uptake versus the ATP turnover rate, and it was like, it seemed like they were increasing their nutrient uptake, but not really their ATP. Do you think that they're like taking up the nutrients because they're there and they're kind of bloating and unable to use it, or what's the story there?
yeah, so that's one of the, with all these other things that we measure in those temperature experiments, like we measure the phosphate uptake and the energy, and then we measure cell counts and particulate phosphorus to see if, with that rapid phosphate uptake, are they actually growing? And that's where with that like low energy rates, it looks like no. And I think that is, if I'm remembering correctly, that is what pans out in the biomass and cell counts, is that they're taking up phosphate, but it's what gets termed luxury phosphate uptake. Like they're taking it up, but they're not using it. They're not actually growing with it. And that is different between the summer experiment and the winter experiment. And that's, interpreting all of the data is ecosystems are complex. So I think it does boil down to that more indications of growth in the winter rather than the summer, and more of this like luxury heat uptake. Like they're taking it up and not turning it into biomass. Yeah.
Kim, thank you for a great talk. You talk very fast, but I could follow.
Can we watch the recording?
Ordinarily listened in. And I too learned a lot today, so thank you. I was going to ask, with those Goshen sections, you talked about
a great question. Is like when we look across, so we got a request from Zoom to repeat the question. So when we look across these big transects, the question is like how things are dynamic and variable was one of the points I was making. So how much do those measurements that we make on, say, a ghost ship's transect really represent the basin-wide processes? And the answer is, thankfully, we do know enough. Not that we get to repeat, say, ghost ship's transects often enough, but smaller segments of them, and particularly things like the time series stations. So the Hawaii Ocean Time Series and Bermuda Ocean Time Series, all the different LTER, the long-term ecological research programs, they help give a really helpful sense of how variable are things on shorter time scales, so measuring month to month and year to year in the same place. So that's where, when we look at broad differences, and like what we were looking at in that South Pacific transect was some really broad differences in like temperature and nutrient profiles, and those are pretty persistent, and those are related to relatively persistent differences in ecosystems, like being cyanobacteria dominated or having the feature of the deep chlorophyll max as opposed to like chlorophyll near the surface. So usually, when we sample across a broad enough section, we do have enough data to tell how similar it is. But we do also pass through crazy features like warm core rings, and the water will look super different. So that's where it is really important that we're always interpreting things in the context of all the data we have and looking at our temperature and salinity profiles to know if this station is representative of just on our transect places to the east and the west, or if this is a mesoscale eddy that we're passing through, and this is not representative of that part of the ocean basin. And those do show up. Sometimes one thing that's fun with the phosphate uptake rate is that you get that data immediately on the ship. We have a scintillation counter on the ship when you're measuring phosphate uptake rates. So a lot of I've had that happen when we have gone through like a warm core ring that like we get to report back. We're like, hey folks, everything looked real different in our afternoon cast, and then we like look at the rest of the data and it kind of pans out that way. So yeah, long story short, we have enough information usually to kind of say how representative these are, and those long-term ecological research stations are really key to that.
I have one follow-up question, if I may. So, if you could choose where you went next in terms of the global scale projects you're working on, what would you consider as kind of an important hotspot where you might learn a lot about processes?
Yeah, so polar oceans, because they're globally really productive and changing really fast. So I'm really excited that Claire's going to the Arctic, and she's working on an Arctic data set right now. And then the Southern Ocean. So on an annual basis, those are the areas of greatest productivity and carbon export. They're also extremely variable. It's a real seasonal boom and bust cycle, whereas the oligotrophic gyres are like low and steady. So I think being able to make these same measurements in poles and subtropical regions gives the contrast that really represents kind of the endpoints of ocean ecosystems. So yeah, polar oceans, specifically the sun. So we're going to get to go to the Arctic, and I'm learning how very complicated the Arctic is in terms of water masses. So the Southern Ocean is sort of our other space we'd really love to go, which that A13 cruise that we were going to go on actually dipped down into the Southern Ocean. So, and it's been continuing to get delayed. So we may or may not get out there sometime kind of soon. We'll see.
I have one question. You were mentioning that we're going to use the deep learning model to study the statistical relationship between the different factors that influence carbon fluxes. First, do you have enough data to actually build like a meaningful network? Two, is it too hard to come up with it from first principles now that you have data? And three, would you be able to sort of explain the dynamics that's going on if you were getting results from a network?
Yeah, so one thing, I guess I'm usually inherently skeptical of like, needing to, of just purely the output of a model, needing like wanting to like truth test that against like, does this make sense with what we can see? The building and execution of that model, this is all through my colleague Jeff Bowman at Scripps. So he has a lot of experience using models like this to go from, say, metagenomic information to inferring ecosystem function. So he's been applying these models in time series sampling sites and transects. So we designed the sampling program with the idea to have enough data points to be able to build the model. So otherwise we would by no means be volunteering ourselves or signing ourselves up to collect like 500 data points for all of these parameters. I've told my grad students this numerous times, I'm like, it's gonna be a lot. So even in 21 days on the ship, like we're doing more than one station a day and every station has many parameters. So we're setting out an ambitious sampling goal to have enough data points to actually be able to use them in the model. And then there's some success that people have for taking data like this and then trying to generate dynamic equations. So like George Hagstrom working with Simon Levine at Princeton has been doing a lot of ecosystem function modeling and doing some really cool work with that. So that's one of the other people we kind of chat with about this. So we would say it's in its like really trying to like pull equations out of this is in its like nascent stages, but that's sort of where we're working at.
Yeah, when you get to the Arctic, there have been some utterly horrendous fire situations. The Fort McMurray fire that burned 13 months through a Canadian winter and across 2 provinces. There's got to be some kind of an enormous signature that's going to make. And the other comment was when you have volcanic ash, it is glass basically. And so the solubility might, in a glass matrix might be a little more difficult than smoke particles.
Yeah, absolutely. So for that volcanic ash, the fact that it's mostly, silica that was ejected at high temperatures, I think that explains a lot of why it is probably low solubility. It also points to one of the things that does come up in the literature is like when solubility has been measured, it is really variable across different volcanic eruptions. They have very different properties at like a macroscopic level, just looking at it, and at a chemical level when it's been analyzed for trace metals. So interpreting impacts for volcanic eruptions is in some ways going to be kind of eruption specific, but I think some generalized takeaways of like expect low solubility more often than not. Yeah.
So just a follow-up. So we've been doing like really detailed analyses on the ash from this Caribbean volcanic eruption and the La Palma eruptions. We have material from both. They look like aerosols from 2 different sources, even though they're both volcanic. The La Palma looks like your typical glass. You can even see shards. The La Soufre doesn't look like glass at all, actually. We also have been using techniques like XPS to look at like what chemicals are on the surface. And the La Palma has like orders of magnitude more phosphorus on the surface than the La Soufre. Trace metals look different. So that's the next step, is to do incubation with the different volcanic ashes and just see if the 2 are similar, and I bet they're not. And none of the models have that. Yeah, that's the next step.
Yeah, exciting things for the atmospheric deposition. Source matters enormously, including within different volcanic eruptions look really different. So lots of exciting opportunities there. Yeah, and the volcanoes have been identified in the paleoclimate records as playing a really important role in changing climate processes on a large scale. So that's a lot of the motivating interest in looking at volcanic deposition. Well, thanks so much, everybody. I don't see any questions.